Hello, welcome to this clinical trial information session about ARO-ATXN2-1001. And just a reminder for folks, because this webinar is being hosted by the National Ataxia Foundation, that anyone participating in a clinical trial or trying a new medication should discuss this matter with their physician. The National Ataxia Foundation does not endorse or recommend any particular studies or treatment options. And with that, it is my absolute privilege to introduce our speakers with you today. We have two speakers and a third who will be joining us for the Q&A session. The first is Dr. Sharan Srinivasan from the University of Michigan. Dr. Srinivasan is an assistant professor within the Movement Disorders Division of the Department of Neurology at the University of Michigan. He attended Northwestern University for his bachelor's degree and obtained his combined MD Ph.D. at the University of Michigan. He then completed his residency in neurology at the University of Michigan before pursuing a movement disorders fellowship at Mass General Brigham at Harvard. He returned to the University of Michigan in 2022 to continue his clinical and scientific efforts in caring for and treating cerebellar ataxias. Clinically, Dr. Srinivasan co-directs the University of Michigan Ataxia Center of Excellence and is the University of Michigan Site PI for the Clinical Research Consortium of the Study of Cerebellar Ataxias. His research interests involve the pathology of cerebellar ataxias, particularly hereditary polyglutamine disorders like SCA2. His passion is in developing novel therapeutics via multidisciplinary approach, combining strategies from organic chemistry, molecular biology, neuro-circuitry, and emerging genetic technologies. His focus has been on targeting impaired neuronal circuitry within these disorders and the amelioration of which has been shown to improve motor symptoms and delay degeneration. So he is our first speaker. Our second speaker is Dr. Massimo Pandolfo from McGill University. He is a professor of neurology at McGill University and the medical director of the Clinical Research Unit at the Montreal Neurological Institute. He earned his medical degree and completed his neurology residency at the University of Milan, followed by an academic appointment at Baylor College of Medicine, University of Montreal, and Université Libre de Bruxelles, where he also served as chief of the neurology service. A fellow of both the American Academy of Neurology and the European Academy of Neurology, Dr. Pandolfo is internationally recognized for his work on ataxias. In 1996, he led the team that discovered the gene responsible for Friedreich's ataxia, a breakthrough that shaped the field. Since then, he has remained at the forefront of basic translational and clinical research on Friedreich's ataxia and other hereditary ataxias. Dr. Pandolfo leads multi-investigator translational initiatives and serves as the principal investigator in several clinical trials. His current work focuses on advancing innovative therapies for neurodegenerative diseases, with a particular emphasis on the clinical trial design and the development of advanced neurotherapeutics. And last, but certainly not least, we have one speaker who will be joining us for the Q&A portion of the talk today, Dr. Gregory Sheckman from Arrowhead Pharmaceuticals. Dr. Sheckman is the medical monitor for the ARO-ATXN2 study and a medical director for Arrowhead Pharmaceuticals. He obtained his medical degree from the University of California, Los Angeles, and completed both his residency in internal medicine and fellowship in nephrology at the Ronald Reagan Medical Center at UCLA. And with that, welcome. I will turn the floor over to our excellent presenters today.
Good morning, everyone, or at least morning for me. Thank you, Celeste, for that wonderful introduction. My name is Dr. Sharan Srinivasan. As Celeste said, I am an ataxia neurologist at the University of Michigan in Ann Arbor, Michigan. And I lead our ataxia program here. And I am here today to talk to you about Spinocerebellar Ataxia Type 2, to give you a clinical introduction about the disease, the pathogenesis, and some therapeutic strategies that are currently being investigated. Just to give you my disclosures, so Arrowhead is offering to compensate me for this presentation. I am not actually an employee of Arrowhead. I have no vested stake in any of the technologies that are going to be discussed today. My job is to give you a clinical introduction about the disease itself. So we'll start with an introduction to spinocerebellar ataxias as a general. So you will hear us talk about them as SCAs or SCAs. Either acronym or shortening is certainly accepted. So these are a group of hereditary neurodegenerative diseases. And there are now over 50 SCAs that have been discovered. And despite these unique genetic causes, many of them share very similar clinical symptoms, several of which are listed in this diagram on the right. And so that might include things like slurred speech, trouble with your eye movements, difficulty swallowing, certainly trouble with your balance and difficulty walking, tremors, and problems with your coordination. These are progressive diseases that are typically fatal, and at the current stage are incurable. Now, specifically delving into Spinocerebellar Ataxia Type 2, or SCA2, we'll get into a little bit more into the genetics, but this is caused by a genetic mutation in the ATXN2 gene, which leads to a buildup of a toxic protein. The clinical features are really characterized by cerebellar ataxia. And ataxia, just to clarify, is not a specific diagnosis so much as a clinical symptom. So when we as a neurologist say ataxia, we really mean lack of coordination, imbalance, not performing things in an ordered kind of logical fashion. And people with SCA2 can have difficulty with their eye movements, oftentimes have slurred speech, and sometimes difficulty feeling their feet or numbness in their legs. The disease often presents in early to mid adulthood and progresses over time. So people gradually get slowly worse over time with loss of function of their motor function and sometimes even cognition as they get into later stages of disease. Globally, each individual SCA tends to be a rare disease, and SCA2 is no different. It affects about 1 to 2 patients per 100,000 people. There are areas where the disease is a little bit more frequent. So for example, there is a specific region in Cuba where there's a much higher prevalence of SCA2 patients, where it's about 40 times the standard prevalence around the world. And typically patients are becoming affected with symptoms in their 30s or 40s. There are certainly cases where patients are affected starting much younger, even in their teenage years. Those patients typically have much larger repeat expansions, which we'll get into in a minute. But it's really important to understand how this disease is impacting folks globally as we plan for both studying and treating these diseases and patients overall. Great. So we're now going to get into the genetics and kind of how the disease actually starts causing problems in our brains. So as I've suggested, SCA2 is a hereditary or a genetic disease. And so our DNA is made up of 4 letters, and that's A, C, G, and T. And there are patterns created by those letters throughout all of our DNA, or called our genome. And in this case, we're looking for a pattern called CAG, CAG, CAG. And that CAG pattern repeats itself, and we count how many times that CAG repeats, and you'll oftentimes hear the term repeat number. And so within the ATXN2 gene is this CAG repeat. And when it's below a certain threshold, there's no problem, you're a normally healthy individual. And when that repeat number expands past a certain threshold, you get SCA2. And that's why this disease is also called a repeat expansion disorder. And so healthy individuals with less than 24 CAG repeats or so, whereas once you have 34 or more, you tend to have SCA2. There is an intermediate range between about 25 and 33 repeats, where patients may have milder symptoms for SCA2, full symptoms, but much more slowly progressive, or actually at risk for another neurological disease called amyotrophic lateral sclerosis, or ALS. CAG encodes for a protein building block called glutamine. And so if you have multiple CAG repeats, you'll get multiple glutamine building blocks tied together. And so sometimes you hear this called a polyglutamine disease, and that's all that means. Glutamine itself is kind of like a clumpy building block that likes to stick together. So when you get polyglutamine stretches of proteins, the proteins tend to clump together, and that can be problematic for the cells in our body and impair the way they normally function. And again, the CAG repeat number is really important in disease. And so on the left here, we're showing a graph where on the x-axis is the number of CAG repeats that are detected. And on the y-axis is the age in onset, meaning the age at which disease starts. And so the obvious thing to take away from this graph is what we call an inverse correlation of repeat length and age in onset. What that means is that the longer the repeat length, typically the earlier the disease starts, and certainly vice versa. And there are a couple of things that are worth taking away, which is, for example, at very high repeat lengths, it's predicted that age of onset will be much younger, for example, even in 20s or in teenage years. But the other thing that I want to point out is that there is tremendous clinical heterogeneity amongst our patients, meaning that even people with the same repeat number can look and present differently. So if you were to draw a vertical line, for example, at 40 CAG repeats at the x-axis, you would see that patients range from developing symptoms at age of 25 to almost 50 years old. So even though there's the exact same repeat number, there's a 25-year difference in their age of onset. So the CAG repeat number is tremendously important, but it doesn't explain the entire story or explain every part of the clinical presentation for SCA2. When we look at the actual pathology of these patients, what we see is that in SCA2, these are what we call degenerative diseases, meaning that the brain is unfortunately slowly shrinking over time. And specifically in SCA2, that's in an area of the brain called the cerebellum, which is the back bottom part of our brain, the brainstem, which is the stalk that connects the spinal cord to the top part of our brain, as well as some of the spinal tracts itself. And this really impairs motor control and how we control our arm movements, our speech, our eye movements, our walking. And we can look even closer under a microscope and see specific cell types and clumping of proteins and loss of specific cell types. And this is really important because when we pursue therapeutics, we want to understand what's the effect of any sort of drug or intervention, and are we not only improving clinical symptoms, but actually preventing cells from dying? And in theory, that would improve the mortality and the lifespan of patients. So it's really important to understand this neuropathology as well. All right. And that leads us to understanding and thinking about these therapeutic approaches. And on the next slide, as I've previously suggested, unfortunately, right now, these are, as they stand, incurable diseases. There are no FDA-approved therapeutics for any spinocerebellar ataxia, and a majority of my time as a clinician with these patients is spent trying to improve clinical symptoms. And that might be treating things like stiffness or what we call spasticity. Patients with SCA2 sometimes have symptoms that are reminiscent of Parkinson's disease, actually. And so many of my patients may get similar medications to help with that Parkinsonism. You might be treating things like urinary dysfunction or leaking or incontinence. A big part of what I do is counsel on the importance of physical activity and cardiovascular exercise and staying active. That means also involving people and specialists like physical therapy, occupational therapy, and speech therapy to practice these tools and keep yourself as active and as mobile as you possibly can be. It's really important to try and be proactive rather than reactive in these diseases, and that means trying to prevent falls. So whether that's having an adaptive device like a cane or a walker, or making sure that your home is safe, removing area rugs around the house, making sure furniture is laid out in a pattern where you can hold on to things as you navigate, railings on stairwells, grab bars, and showers are really critical to making sure that we keep people safe. And anytime I hear anything problematic with swallowing, it's really paramount to make sure that we stay on top of that and evaluate the risks and implement strategies to try and make swallowing safer. And the last thing that I've listed at the bottom is family planning. I'm a firm believer that diagnosis and having an established diagnosis of a disease can be really therapeutic. And at least in my experience, many of my patients who come to me with ataxia are on this diagnostic odyssey and on a journey to understand what's going on. And having a definitive answer is really therapeutic and cathartic for them in understanding their disease, understanding what to expect, and being able to convey that to both family, friends, and other loved ones. But as you'll hear much more about, there are many of us who spend our lives trying to study these diseases in hopes of doing better. And so understanding the basic science that drives this disease is really critical if we're to try and uncover novel drug targets and novel therapeutics to help treat clinical patients. And so I've listed several of the current therapies that are being studied right now, including one that many of you may have heard about, a molecule called troriluzole, which is being explored for treatment of multiple different SCA types and is being reviewed by the FDA currently. There is a recombinant small molecule, a small protein called neuro-EPO, which is supporting the growth of our brain cells or neurons and hopefully helping them communicate better with each other. And then a drug approach that you're going to hear more about, which is called an siRNA, or a small interfering RNA, that will help decrease the expression of the harmful toxic protein that is expressed in these diseases. And there are other genome editing approaches that are being explored to try and help correct the underlying genetic mutation. And if you're interested in hearing more about other drugs that are in the pipeline, you're welcome. And I put below here the website. The National Ataxia Foundation has a wonderful drug discovery pipeline for all of their SCAs. And you can visit that website below to get more information. But more specific to this presentation, since this is a genetic disease, one of the strongest approaches to trying treating this disease as best as we can is to target that genetic mutation. And so since we know that the repeat expansion, that CAG repeat pattern that we described earlier, causes buildup of this toxic protein, one strategy is to try and reduce expression of that toxic protein. And so that's what this siRNA technology, or some of you have heard of what are called antisense oligonucleotides, or ASOs, or gene silencing strategies to try and prevent these toxic mutations from being expressed and causing harm to the cells in our body. And there's a wealth of data showing that if you do such a thing, if you reduce the expression of that toxic protein, that the cells go back to being much more normal and they're able to function the way they're supposed to. And that animal models and larger animals show improved neurological outcomes, and they're able to have improved motor function. They're not ataxic, and even slow down the disease and improve mortality in some cases. And so that brings us to, we're currently in the phase of therapeutic discovery and development. And we can't know whether the drugs that work in our mouse models and even in our larger mammal models will work in a human, because at the end of the day, nothing replaces a human. And a mouse is not a human, a primate is not a human, and there are differences in our biology that make clinical trials absolutely essential if we're going to bring new drugs to market and be able to treat and help patients. And so it's really critical that we have these trials that we can answer these questions, and what works, what doesn't work, and what matters to a patient, what symptoms are actually important to you, what biology is actually relevant when it comes to treating a disease, and what biology can we stand to live with. But ultimately, the goal is really to remember the goal here is to get treatments to our patients. And so everything we do has to be done with that mindset. And so just to wrap things up, I think, I've covered a lot of things, mainly that SCA2 is a genetic disease. It's driven by a particular repeat, a pattern that expands, and that we can detect this, and that there are strategies to try and reduce expression of that toxic gene product, that toxic protein, and that is a viable and a potentially very helpful way to intervene in patients and help them get better. But we need more information, and we need to use clinical trials as best as we can to help not only treat our patients, but to learn how best we can be moving forward. And so with that, I will, I think, hand it over to Dr. Pandolfo.
Thank you very much. And My name is Massimo Pandolfo. I am currently professor of neurology at McGill University, and I am the medical director of the Clinical Research Unit at the Montreal Neurological Institute, which means that I'm very much involved into what we call translational research, that is trying to bring findings from basic science to fruition as in the clinic as potential new therapeutic approaches, particularly for conditions that had no previous that have currently are incredible, incurable, as it was said by Dr. Srinivasan and concerning SCAs and SCA2 in particular. Welcome to the seminar, and good morning and good afternoon and good evening, considering how much you are scattered throughout the world. May I have the next slide, please? These are my disclosures, and for this presentation, I'm the principal investigator for this trial at the Montreal Neurological Institute, and I may be compensated for this presentation. I'm saying that because I don't want to create any potential conflict of interest with my position as principal investigator. Let's move to the next slide. So what I'm going to do in the next 10-15 minutes or so, I will tell you about this study called the ARO-ATXN2-1001 study, which, as Dr. Srinivasan has said, is aimed to reduce the level of this toxic protein, which is the mutated ATXN2, the form of ATXN2 that contains a too long stretch of glutamine, the so-called polyglutamine protein. Now, this slide is taken from a from a website called clinicaltrials.gov that has basically, where basically all clinical trials going on throughout the world are listed. And of course, this site carries some synthetic information about each of this trial. I will just, gonna get through all of this a little bit more in detail and in a clearer way that this very specialist directed way as you can find in clinicaltrials.gov. But let me go through the official trial title of the study. This is a phase 1 placebo-controlled dose escalating study to evaluate the safety, tolerability, pharmacokinetics, and pharmacodynamics of ARO-ATXN2 in adult subjects with Spinocerebellar Ataxia Type 2. Let's go to the next slide, where all this kind of exotic sounding words are explained. What is a phase 1? What does it mean, phase 1? Phase 1 is when a new treatment is given to humans for the first time and is essentially aimed to evaluate whether this treatment is safe. As it was said before, animal studies precede phase 1. They should reassure us enough that the drug doesn't carry excessive risks of toxicity, and there are clear hints that it may be helpful. But we need to get into humans, and that's where we get into humans for the first time. Placebo-controlled means that participants in the study would be randomly assigned to receive either the active drug or what is we call a placebo, which means something that looks like the drug. In this case, it's a liquid. The drug comes in a liquid form inside a syringe, and it's a syringe that contains a liquid that looks exactly like the drug, but does not contain the drug. In fact, is a completely inactive product. It's dose escalating, which means that we will start the study with a dose that we expect to have an effect, but is the lowest dose that we expect to have some kind of effect in humans. And we will check whether that is safe, Then, if that is safe, we move to a higher doses that may be more effective in principle, we don't know, and we check whether that is safe. And again, in this particular study, we will see we move through 4 different cohorts of participants, and therefore through 4 different increasing doses. Studying pharmacokinetics means what happens to the drug once it's introduced in the body. Every compound introduced in the body, it's somehow eliminated in a way or another. It may be degraded by specific, for instance, by the liver, but also by other cells in the body, and it may be eliminated, and it's eliminated with time. So we want to see what happens if you introduce a certain dose of the compound in the body, how long it stays there, how long it takes to decrease to half of the initial dose, what we call its half-life, and what kinds of concentration it gets in the various body fluids, like in this particular case, in the cerebrospinal fluid. So this is very important to determine, together with the other data, what is the best dosing for this drug, how often we need to give it, and how much of it. And pharmacodynamics is essentially to check whether this compound is doing what it's supposed to do. So this thing is supposed to decrease ATXN2 levels. Well, we're going to check whether it really does what it's supposed to do, and how this correlates with how much of the drug we give. So that's another very important information that we need in order to establish if this drug turns out to be safe and effective, what is the best way of giving it, how much and how often. May I have the next slide, please? This particular study, therefore, is following the criteria that I just mentioned, is structured into 4 different cohorts. In each of this cohort, basically 2/3 of participants will receive the active drug, and 1/3 will receive placebo. The 4 cohorts, so the first cohort starts with the lowest dose, and it's a small cohort, it's just 6 participants, 4 with the active drug and 2 with placebo. The first 2 participating patients will be carefully checked to make sure that there is no safety signal and nothing that looks like that the drug is not toxic and we don't need to proceed. If the first 2 patients, and this holds for all the 4 cohorts, seem to have tolerated well the drug, then the rest of the cohort is given the product or the placebo. There is a safety review, and at that point we move to the following cohort. The second has a little higher dose and is again just 6 participants, and then higher dose in the third cohort that has at this time 12 participants, and the highest dose in the fourth cohort that has 12 participants. So this is open to recruitment, is still going on, and in a number of sites throughout the world. May I have the next slide, please? So what is the product that we are going to inject? That has been already mentioned by Dr. Srinivasan, but I'm afraid I barely mispronounced his name. But let me tell you a few more words about what is ARO-ATXN2. This is what we call a small inhibitory RNA, or siRNA. This is really a clever way of using a mechanism that our cells have to control the expression of genes. What does that mean? You know that all cells in the human body carry the whole information to make a whole human body. But of course, this is not used all at once, and it's not used and each cell doesn't use all of it. Clearly, during the body development, we need to use part of the information to build up the body. And then, of course, a brain cell is not going to use the same information of a liver cell because it needs, it's made differently, it operates differently. So cells contain a number of mechanisms that turn on and off parts of the genetic information, and siRNAs are one of these. RNAs are basically copies, slightly different, made by a slightly different molecule, which is called ribonucleic acid instead of the deoxyribonucleic acid, RNA instead of DNA, of the genetic information. And these RNAs, they have been known for many decades to carry the information to make proteins and therefore to make the building blocks of cells and the molecules that make a cell function, work. But some RNAs do not do that. They have a regulatory function. And this inhibitory RNAs, what they do, they are made by certain cells that this way repress the expression of genes that are not important for those cells. And so what is the clever use of this mechanism? Well, let's make one of these RNA that represses the expression of the ATXN2 gene, because that's making a toxic protein and we want to turn it off. And this has been tested in animals. It works in animals, so we expect it to work in humans as well. And we have seen that even higher doses that we plan to give to humans have been tolerated well by animals, so it doesn't seem to be toxic. There is often a question at this point, we have two copies of each gene, and in the case of people with ataxia with SCA2, one is the one that makes the toxic protein, and the other one makes the normal protein. Is this thing going to decrease the level of both? The answer is yes, but this seems to be very well tolerated in animals, and even animals, mice that do not at all make any ATXN2. They are viable. They grow up. They reproduce. They are a little bit big, but they don't have any very serious consequence. May I have the next slide, please? So this compound, this product needs to be given directly into the liquid in which that surrounds and fills up the nervous system. This is called the cerebrospinal fluid. It surrounds the brain and the cerebellum. It's contained also in some what we call the ventricles, that are cavities inside the brain, but it also circulates down along along the spine all the way to the lumbar spine, so the low back. And that's where it can be, I would say, easily accessible by what we call a lumbar puncture. This procedure implies to introduce a needle that you can see in the image here. It goes through between the vertebrae all the way to the what we call the dural sac, so this part that is contained within the spine where the liquid is found. And then the product is injected directly there, so it can go directly in contact with the brain, with the cerebellum, with the brainstem, those structures that you have learned are affected in this disease. It's not just the RNA itself that it's injected, it's packaged into a special package made of lipids in order for it to be protected and delivered properly to the cells where it's supposed to go. A lumbar puncture is something that some people are a little bit worried about. Again, it's done with I personally, when I do it, I generally use local anesthetics. So I had really no patient during the procedure that complained that I had pain while the procedure was performed. At most, they feel like a mosquito bite when the the needle containing the anesthetic essentially is introduced, most of the feeling on the side for the patient is at most a feeling of pressure in the back. It's really a low-risk procedure. It's done at a level where the spinal cord is no more present. There are just nerves descending into this fluid, but they are sort of put aside by the needle. The risk of damaging these nerves is really extremely low. And there is no risk of damaging the spinal cord because it's up and above the level where the injection is made. We use a very thin needle to do this in order to prevent the fact that there may be a leak of fluid in the trajectory of the needle once it's extracted, and this causes headache. Now, the thinner the needle, the less likely this leak of fluid is, and that's less likely is that that the person that has had the procedure may have headaches afterwards. I mean, this doesn't mean that the risk of having a headache is zero, but it's low. And in most cases, these headaches is just mild. So that's we try to do in the safest and the best tolerated patient with the maximum comfort, or you might say minimum discomfort for the patient. And so this is, we also use certain technical, let's say, procedures that allow us to reduce the time for this procedure to probably 10 minutes or so. So it's really not a tragedy and nothing to be scared about. May I have the next slide? So who can participate? Very, very quickly, let's go through this study. This study, includes only adult patients. This is normal. Usually when something is tested first in humans, is tested in adults. In children, only at a later stage. And also there are not many children with SCA2. So this is up to 65 years of age in order to avoid individuals that may have, as you get old, additional conditions that can make participation in the trial less safe. They needed to have a clear diagnosis of SCA2 with at least 33 of these CAG repeats. And this can be based on medical records or genetic tests that can be done at screening. There is a particular scale, I will say a few words about that tells us how bad an ataxia a person has. The higher the score in the scale, the worse the ataxia is. It has to be 14 or lower, which means it has to be someone who is still able to walk without support. Essentially, his coordination problems are still limited. And this is because, again, we want to see these are the patients that are most likely to benefit from this kind of treatment when there is not too much compromise already, that pathology that you have seen, that shrinking of the cerebellum and the brainstem is not too advanced. We cannot include people who are taking another investigational therapy, and I think that's for obvious reasons. It would mess up things horrendously. And certain medical conditions also are exclusion criteria in order to protect the safety of participants. And the BMI of more than 40 means people who are very large. That can make the procedure of the lumbar puncture difficult, more complicated than what I just described. If there is a thick layer of fat between the skin and the spine. And of course, there are some conditions, for instance, conditions that decrease the ability of the blood to clot, to form clots, that are contraindications for the lumbar puncture, or a damage the spine, that that can really make the procedure technically difficult. So these are the main inclusion and exclusion criteria, but all this is clearly reviewed by the study site with any prospective participant. The next slide, please. So there is a visit called the screening visit, in which eligibility is determined based on this criteria. There is a review of medical history, measurement of vital signs. There is a physical neurological exam. There is the assessment of the scale called SARA, an electrocardiogram, blood and urine samples, and an MRI scan, which essentially is aimed to show that there is no other neurological problem, condition that may cause similar symptoms, so that is really the participant has SCA2, but his brain is not otherwise affected. May I have the next slide, please? And then the study period begins if all inclusion and none of the exclusion criteria are fulfilled. The study starts. The first day is given a lumbar puncture in which some fluid is taken and the product is the study drug is injected. And again, there will be a number of exams and physical neurological exam, electrocardiogram, blood tests, etc. In particular, is necessary to make sure that coagulation of the blood works fine to avoid the risks of bleeding from the lumbar puncture. And then there will be a number of follow-up visits, including 4 total lumbar puncture over the course of the study. And these are indispensable to study, you remember, those pharmacokinetic and pharmacodynamic things that we mentioned before, to see what happens to the drug, but also to make sure that there is no reaction, unwanted reaction in the central nervous system, like inflammation, for instance, some kind of toxic reaction to the drug. So it involves safety, but also involves those other parameters that I mentioned before. May I have the next slide? Now, just a few words about some of the tests to check the severity of ataxia symptom. The scale for assessment and rating of ataxia, the SARA, was developed about 20 years ago for a study that was called the EuroSky. I was one of those who developed that scale. It was a European study of many of these spinocerebellar ataxias, and it basically tells you how bad your ataxia is, with 0 being no ataxia at all, and the highest score is 40, meaning at that point they cannot even perform the test because the symptoms are too severe. There is an inventory of non-ataxia signs, which is basically a list of other problems that a person with spinocerebellar ataxia may have, like, for instance, visual disturbances, sensory disturbances, stiffness, etc., that are not considered in the scale for us, in the SARA scale. And then there are a number of standardized tests, like the 9-hole pegboard test, so to see how long does it take to put those pegs into 9 holes. You can see the image there, how it works. Of course, if you have ataxia, it will be more difficult and it will take more time. And the click test, you have alternative click on the those two. What are those? They look like the counters, like for cell counters. We use that, but you click on those 2 buttons, and again, it's assessed how long does it take to click 10 times each of these buttons. There are questionnaires that inquire about quality of life, health-related quality of life, the type of difficulties and uncomfort that people may have, the loss of well-being that people have because of ataxia. And there is this global impression of change that is asked. That how do you feel compared to before? You think you feel the same? You think you feel a little better, a little worse? and this is really, what we call the patient-reported outcomes. That is what the patient really has to say concerning their function and their well-being. May I have the next slide? And of course, participants should attend all clinic visits, complete all scheduled procedures and assessment. They have to make a commitment to do that. They cannot participate in other clinical studies. I mentioned already that would create so much confusion that the data would be not interpretable. It's important to inform the study physician about any changes in health, any side effects. When we do a clinical trial, so we collect everything that happens to participants. We call them adverse events. But even if you fall from a bike, even if you, I don't know, if you get the flu, whatever, anything that has affected your health needs to be recorded. We don't know in advance whether that's related to the study drug. In some cases, it's obvious that it's not, but nevertheless, we need to have a complete list of everything that happens to participants. This is extremely important in the end to assess the safety and to detect maybe even unexpected problems that have appeared during the study. So this is very important. And since we don't know what this compound can do on embryos, you should participants should must actually agree to use highly effective contraception during the study and for at least 90 days after the study. This doesn't mean that this product is toxic for the embryo. Just means that we don't know, and this is something that can be considered at a later time if this thing seems to be safe and effective, otherwise safe and effective, but not at this time. May I have the next slide? The primary endpoint, the conclusion that we're going to get from this study, whether this thing is safe and tolerable. I just mentioned we needed to identify the side effects, even unexpected ones. And then we want to know something about the pharmacokinetic, how the body metabolizes and get rid, how long does it last in the system, and if there is any untoward reaction in the central nervous system, as I said, for instance, a reactive inflammatory reaction that can be dangerous. The next slide, please. And of course, we want to see whether this is the famous pharmacodynamic aspect. Does this succeed in decreasing the level of ATXN2 protein, which is what we want to happen? And that's something that's why we do repeated lumbar puncture, not only to see what happens to the drug once it's injected, but also what happens to ATXN2 and what happens to other markers of degeneration of nervous cells, like this thing called the neurofilament light chain, which is basically a product that comes from degenerating nerve cells. If there is ongoing degeneration of nerve cells, this neurofilament light chain will be more abundant. If neurodegeneration slows down or stop, it will decrease. So this is an exploratory thing. Is this thing doing anything to the neurodegenerative process? And then we have those clinical function measures that I just mentioned. And finally, the MRI will tell us whether there was any shrinking of the brain that happened during after this treatment, and how this differs from what would be expected in the progression of this condition. MRI, of course, also checks for safety, for instance, again, for any sign of untoward inflammatory reaction in the brain, any damage that might have been triggered by the drug. And so it has a double, both efficacy and safety, and again, mostly safety objective. Next slide, please. These are the sites. We have 2 sites here in Montreal, and the sites are, as you can see, throughout the world, but not in the US. Nevertheless, we have been able to accommodate patients from the US here at our site, at our Canadian site. So patients from the US can participate in the study. They just have to travel a little bit to come over here, have a passport, and have an insurance that will cover anything that may happen that is unrelated to the study while they are here. Still the end of the summer, but in the winter we have ice on the street. If they slip and fall and break a leg, they must have some kind of insurance to protect them. But they are very welcome to participate. And of course, for anything that is related for the study, all patients are insured. May I have, do we have any other slide? Or yes, you can learn more here. There are some links that you can use to learn about this and other clinical trials. And of course, you can have a sponsor contact at Arrowhead that also can be a source of information if you're interested in this trial. Thank you for your patience and for listening to me. Questions?
Wonderful. Thank you so much, Dr. Pandolfo and Dr. Srinivasan, for this fantastic presentation. We are going to move into our Q&A session. Just a reminder for folks, if you have questions, so please type them in the Q&A window, which you can find at the bottom of your screen. We'll be jumping right into questions and getting through as many as we can before the top of the hour. And we have both Dr. Pandolfo, Dr. Srinivasan, and Dr. Sheckman from Arrowhead, who is on the line to answer questions. The first one is, you mentioned this is a phase 1 clinical trial. What would the timeline be for moving through this trial and then getting it towards something like an FDA approval or another regulatory approval?
I think Dr. Sheckman is probably the best person to answer this.
Sure, thank you. And thank you for that question. So as both Dr. Srinivasan and Pandolfo mentioned, this is a phase 1 clinical trial. So at this stage, we're really first bridging the gap between the preclinical data that was generated in animal models and evaluating the safety of this drug in humans. So at this very early stage, it's very hard to put a timeline on things like FDA approval or things like that, or commercial availability, because drug development takes a long time, and this is why it's such a complex process, but it has multiple steps. There's usually these very early phase 1 trials that evaluate safety. Then there's also phase 2 and phase 3 trials, after which approval can usually take place. So it's very early at this stage. We're really just trying to understand the safety tolerability of this compound, identify whether there's evidence of benefit, and that would really allow us to take this compound into later phase testing and move towards approval. But timelines for that are pretty hard to provide, unfortunately.
And then another question from an attendee. If you become a participant in this trial, is travel paid for?
Yeah, so that's a great
Yes.
question. Do you want to talk a little bit about sort of your experience specifically?
Sure. Yes, the travel is arranged and is paid for. So you can, accommodation, anything that is required to come over here and to spend the time here. When we give the drug, for instance, you are not going home right away. You have to stay overnight. And if you are coming from far away, you may need to stay for instance, one more night or so. So there is, everything is taken care of.
Yeah, and I will maybe add to that slightly that if you if you have a caregiver, travel for the caregiver can also we can also make some accommodations for that, and that's something you can discuss with the study team at the site you reach out to.
Good to know.
Yes, absolutely.
And then Dr. Sheckman, would you mind sharing your screen again, just for the contact and resource screen? We've had a few people ask if that could go back up on screen so they can take a quick screenshot of it. If you do miss that information and would like to have it, also feel free to email research@ataxia.org. We can share that with you.
Hopefully that works okay.
Fantastic. We'll just leave that up there when we're going through the rest of the Q&A. We have time for a few more questions. We're running out of time here. Another question from an attendee. Has recruitment started at all the open centers?
Not at all open centers, but it has started.
Yeah, so all of the centers on the left column, those centers are recruiting, and I can maybe go back up one more, I think. So everybody on the left is open and recruiting. The sites on the right are sites that will be open, hopefully soon.
All right, if you wouldn't mind going back to the other slide, just so that people can take that screenshot if they missed it. And we have time for two quick questions. If this drug were to be successful and approved, then could you remind us how often it would need to be administered or delivered to patients in order to be effective?
Yeah, that's a great question. So one of the things we are trying to learn as part of this phase 1 trial is exactly that. So the measurements that Dr. Pandolfo was mentioning, the pharmacodynamics specifically tell us once this drug is administered, how long does the effect last? How long is the reduction in that abnormal protein detectable for? And that would then allow us to better characterize the frequency at which it would be administered, and that would be studied in later trials. Thank you for that question.
Very good to know. Thanks for clarifying that. We have time for one last question. If you could just clarify again, if people are interested in signing up, what's the best way to go about that? Should they be going to the clinicaltrials.gov page? Should they be emailing you? What's the best way to start that conversation?
Yeah, that's a great question. So any of these will sort of get you to the right place. So the clinicaltrials.gov link has a list of sites, which often includes contact information. However, please feel free to utilize that sponsor contact email. We've already had patients reach out to us through that venue. That inbox is monitored. We can put you in touch with the site that is closest to where you reside or where you would like to participate, and put you in touch with the study team at those sites, and the sites can take it from there.
And we are at the end of our talk today, but we weren't able to get to all questions. If you do have any additional questions, please feel free to email the Arrowhead team at ARO-ATXN2@arrowheadpharma.com. Or if you're forgetting that email, please feel free to send them to naf@research@ataxia.org. We'll forward them on to the team. We'll make sure they get to the right person. Dr. Sheckman, Dr. Pandolfo, Dr. Srinivasan, thank you so much for joining us today and telling us more about this clinical trial opportunity. We really appreciate your time and expertise.
Yes, and thank you to all of the patients that joined the webinar. We really appreciate your time as well, and hopefully this was informative for you as well.
And a copy of this webinar will be made available on the NAF YouTube page in about a week. If you missed something, don't worry, it will be shared out there. And I hope that all of you who are attended here today, you learned something new about this clinical trial and maybe learned something new about SCA2 overall. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, everyone. Bye.
Thank you.